Hey there, anatomy students, and welcome to the first episode of our video lecture series. Hopefully this will be a complement to your studies. Since we're not going to be meeting face-to-face, I think communication is going to be key to maintain. So please email me with any questions or comments you may have. We'll hopefully have our discussion board up and ready to go really soon. So that we can maintain some level of interaction, which is, what makes class time so awesome is that we have real-time interactions, which are not possible this way, I know, but hopefully this can, again, help to supplement your learning. So keeping up with your readings in the books, doing the activities that I assign online either through Momentum or on the Pearson Mastering, keeping up with that is going to be critical to facilitate your learning. So today what the content of this lecture is going to be about is kind of a basic introduction to metabolism, and particularly we're going to be emphasizing where energy requirements of the body are met. So how does the body produce the energy currency, which of course is ATP. Where does ATP come from? So we'll start off just talking about metabolism more generally. Metabolism can be broken down into two fundamentally different types of reactions. Metabolism is all of the chemical reactions going on in your body. But you can break those down into either anabolic, let me get a marker that works, either anabolic reactions or catabolic reactions. So what is the difference between these two? Well, the fundamental difference between them, the primary difference, is that anabolic is going to be synthesis reactions, where you start off with discrete monomers. So here I've drawn 3 monomers of some substance. So these molecules, these could be glucose molecules. Any monomer, these could be nucleotides. You start off with a monomer, and then what you synthesize is the polymer. So in the process of anabolism, you're taking monomers and you are covalently bonding these monomers to one another. So these monomers are now sharing electrons, and that requires energy. So there has to be an energy input before this reaction will proceed. This does not happen spontaneously without the input of energy. Where does that energy come from? Probably jumping ahead a little bit, that's ATP. But where do we get the ATP to supply energy for these reactions? And that comes from our catabolic set of reactions. So again, anabolic reactions can be kind of categorized as synthesis reactions. Catabolic reactions are going to be the opposite. You're going to be breaking down polymers. So let's draw our little polymer here. We have the opposite. We're starting with the polymer here instead of starting with the monomers. And we are removing these chemical bonds between the monomers that make up this polymer. So in breaking those bonds, we are releasing stored energy. So this is kind of a theme that will continue throughout this lesson, is that when electrons move around, they release energy. There's energy stored in the covalent bonds between these monomers. When you break those bonds, you release energy. So these reactions, anabolic and catabolic, there's a balance of them constantly going on in the body. So if you think about what's happening through development, if you have a child that's getting larger as time goes on, you can probably imagine their body is increasing in size because they have more of these anabolic reactions going on at any given time than they have catabolic. Their bodies, because they're growing, they're favoring synthesis reactions. There's more new, larger molecules being formed than there are large molecules being broken down to provide energy. And that requires lots of energy, and so that's why someone who's growing, if someone's going through a growth spurt, for example, that person's going to be consuming a lot of food, and that's to supply energy that's needed to perform all of these synthesis reactions. So another way to describe these reactions is when you're thinking about what is going on with the energy. So where is the energy? Is it being input or is it being released? So for these anabolic reactions, synthesis, where it requires an input of energy, we would say that those reactions are endergonic. They require the input of energy in order to make those reactions progress. Whereas catabolic reactions, they're going to release energy. And those types of reactions are called exergonic reactions. And when you release this energy, that energy is oftentimes going to be stored in the form of ATP. This molecule ATP, adenosine triphosphate, is going to act as a full battery. It's storing energy that can be rapidly accessed depending on the needs of the cell. So if your cells need to perform a bunch of these synthesis reactions, they're going to be using the energy stored in ATP to do that. And how they build up reserves of ATP is by performing lots and lots of these catabolic processes, breaking down food. So most of the food that you eat is going to be filled with lots of polymers. The proteins that you ingest, the lipids, the fats, those are mostly going to be stored in large molecules that are going to be broken down, and that energy that's liberated when you break down those molecules is used to make more ATP that you can then use for whatever your body needs them for. So just thinking about catabolism right now, what are the types of substances that are being produced by these, that are being used by catabolic processes? You have substances like glucose that can be broken down to provide energy. That's the primary energy source for most of the cells in your body, is going to be glucose. So that's a very important molecule that stores lots of energy in the bonds. So what glucose looks like, more or less, is it's going to be this ring of carbons. So each of these points, remember, represents one carbon, and every line represents a covalent bond. And there's lots of energy stored because there's lots of carbon-to-carbon bonds here that can be broken. Energy can be released when this molecule's bonds are broken. So glucose metabolism, glucose catabolism, importantly, is going to provide lots and lots of energy for most of the cells in your body. Triglycerides. So what a triglyceride is a stored fat. You have fatty acids. Remember, fatty acids have these long tails. And you have those fatty acids being joined to a molecule of glycerol, which is actually related to glucose, which is why the name sounds a little bit similar. So a glycerol molecule that's bound to these fatty acid tails. This is how fats are stored in your body. So in adipose tissue, you have lots and lots of these triglycerides that store your fatty acid tails. What you can also notice if you look at those fatty acid tails, each of those points represents, again, a carbon, and each of those lines between the points represents a covalent bond that can be broken and energy can be released when that covalent bond is broken. So this doesn't happen as efficiently as it does when you break down glucose, but you can still release energy from the breakdown of fatty acids. So that will happen in cases of starvation, or if you're under calorie restriction, your body will shift because you're not taking in as much glucose. Your stored triglycerides are going to be broken down, so you will remove these fatty acids from the glycerol molecule and start to break the bonds between the fatty acid tails, and that can provide some energy. And also your proteins that you ingest can of course be broken down into amino acids. So this is just generally going to be what most of the catabolic reactions in your body look like. You have your glucose that's being broken down, triglycerides, proteins, mostly things that you're getting from your diet. Now, what are we using all that liberated energy to do? We're using it mostly to make ATP. So ATP is produced when you add an inorganic phosphate to ADP. So why does our body produce ATP? Well, it's because ATP is, again, that charged battery that's storing, that's got potential energy that can be used to drive processes like synthesis reactions. So let's say we've got some synthesis reaction down here. You've got your two monomers. You're going to add a covalent bond between them to make some larger molecule. That takes energy. Where do you get that energy from? You get it from taking ATP, splitting that, breaking the bonds between ADP and that inorganic phosphate, and you release energy. That energy can then be used to drive this process of synthesis. So let's go back to where that ATP came from again. So you have some polymer, let's say it's a starch that you ate in your diet. Potatoes are full of this, polymers of carbohydrates. So you break down that polymer, break down some of the bonds between carbons in that polymer, whatever it may be, some catabolic reaction. You're left with those monomers and you release some energy. Or you're left with things like carbon dioxide that are being released. But those catabolic reactions produce energy. And what happens is that energy, once it's released, is going to be used by cells to power the formation of ATP. So here we have this stored energy, this fully charged battery of ATP that can then be used to power whatever synthesis reactions, whatever anabolic processes the body may need. And there's lots of other processes in the body that require ATP. There's those ion pumps. So many needs, many requirements in the body for energy. Many processes that require energy, that is. But in this case, what we have here is coupling of these two reactions. You have your catabolic reactions and your anabolic reactions. And anabolic reactions require catabolic reactions to generate ATP that can then be used to power those. So in this way, we say that these reactions are coupled. This exergonic and endergonic reaction. They rely on one another. The anabolic reactions are going to rely on a catabolic reaction to provide energy, the means for them to make that reaction proceed. So now I want to take a few minutes to talk about movement of electrons, which we've already established is pretty important. Movement of electrons is going to release energy in lots of cases, particularly when you break bonds. But generally when electrons move, they release energy. And there's a specific subset of chemical reactions called oxidation-reduction reactions. So an oxidation-reduction reaction is going to be a chemical reaction where there is movement of electrons. So let's think about what I mean by this. So a molecule that I'll use to illustrate this type of reaction is sodium chloride. This is table salt. So sodium chloride, this sodium is held together with this chloride by an ionic bond. So what's going on here is you have a positively charged sodium that is interacting with a negatively charged chloride ion. So these are not actually sharing, the sodium and chlorine are not sharing a pair of electrons. What they're doing is sharing an ionic bond. They both carry a charge. So what happens to produce sodium chloride? Well, you have sodium, which is not very electronegative. You have chlorine, which is very electronegative. And what's going to happen is an electron, which again carries a negative charge, from sodium is going to be transferred to chlorine. So when you take that electron away from sodium and give it to chlorine, that gives chlorine a negative charge. It's got one more electron than it had, one more negative charge. So that chlorine is now negatively charged. It's taken an electron from sodium. Now when you remove that electron from sodium, because you've removed a subatomic particle that has a negative charge on it, you're taking away a negative, so that makes sodium positive. So here you have had an electron moving from sodium to chlorine. So now where you had two neutral or uncharged atoms, now you've got sodium with a positive charge, chlorine with a negative charge, and now they can, that positive and negative on those two atoms, it's going to make them interact. They're going to be drawn together because, of course, the age-old adage is that positive attracts negative. And so that's what an ionic bond is. But before that ionic bond is formed, there's movement of an electron from sodium to chlorine. And when those electrons move, that is an oxidation-reduction reaction. So an oxidation-reduction reaction, again, is just movement of an electron. And one substance in that oxidation-reduction reaction is going to be oxidized, and another substance is going to be reduced. Oxidation-reduction. So a way that you can remember which is which substance is being oxidized and which one is being reduced, is through this mnemonic called oil-rig. What oil-rig stands for is that oxidation is loss of electrons. So whatever is losing an electron is going to be oxidized. And reduction is gain. So whatever is gaining the electron is being reduced. So in this case, where you have sodium losing an electron, it's being transferred to chlorine. Sodium is being oxidized. and chlorine is being reduced. So transferring electrons is something that happens in many types of reactions, but specifically ones that are being used to trap energy. So when electrons move, they release energy, and that energy can be used to do work. So oxidation and reduction reactions are going to be an essential part of capturing energy in the form of ATP. So right now I want to use a real-world example to kind of illustrate how flow of electrons can release energy and how that energy can be captured or can be used to do work. So think about where you know of where there's flow of electrons. You might think of a battery. So that's exactly what's happening when you connect the two poles or the two sides of a battery, is you're allowing charged particles to move. And as charged particles like electrons move, they can release energy, and you can use that energy to do work. So we mentioned the oxidation reduction reaction. The substance that's being oxidized is losing an electron. So you have this electron that's moving from one substance to the other. So let's say this is a substance that's losing an electron. This is a substance that's gaining an electron. And as that electron moves, it can release energy, and that energy can be used to do work. If you think about what's happening inside of a battery, there's a plus side and a negative side. So let's actually draw that the other way. Say this is the plus side and this is the negative side. And in a battery, you're having these charged particles moving from one end to the other, from one substance to the other. So you'll have on one side a substance that has a high affinity for electrons. So that term affinity just means how attractive it is to electrons. Something that has a high affinity for electron is going to be likely to attract electrons. Now you have this substance that has a low affinity for electrons, meaning it's going to be less likely to attract electrons, and it's going to be more likely to lose electrons. So that's exactly what's going on in a battery. You have one substance that has a high affinity, one that has a low affinity. Electrons bounce from one place to the other. And so how they bounce, or how they move, is that you connect some conductive wire between to connect the two substances. And electrons can move through this wire. So somewhere in the middle of that wire, you're going to have something like a light bulb, or whatever the electricity is being used for. In this illustration, it'll be a light bulb. But as that electron flows through this wire, it's going to be diverted through the filaments in this light bulb, and it's going to give off energy as it does. It's going to be able to do work as those electrons flow through. And that's, in the case of a light bulb is going to use that energy to produce light. Now your body basically does this same process, but instead of generating light, it's going to produce ATP. Your body, particularly the mitochondria inside every one of your cells, are kind of like biological batteries. They're harnessing the flow of electrons to produce ATP. And then splitting that ATP can release energy that can be used to do all sorts of work, like synthesis reactions or pumping a substance against its concentration gradient. So putting a substance where it normally wouldn't be flowing. Whatever processes require energy, you can use ATP to do that. So your mitochondria are going to harness the flow of electrons to produce ATP. One last thing about this battery analogy is that in this case, the low affinity substance that's losing the electrons, because oxidation is lost. If you go back to oil rig, the substance that's oxidized is going to be losing electrons. So this substance that has a low affinity for electrons is going to be oxidized. The substance that's high affinity is going to be the one that is reduced. So the importance of those oxidation reduction reactions is that they're used to carry electrons and move electrons, and as those electrons move, they can give off energy that can again be used to generate ATP. So in this process of generating ATP, you have special molecules called electron carriers. These electron carriers have a high affinity for electrons, so they're going to be more likely to be reduced. They're going to be gaining electrons. And when they gain electrons, they carry those electrons. We'll see in just a few minutes where they're eventually carrying those electrons to. But these electron carriers, the two prime examples of these with regard to how we generate ATP, the two electron carriers that we're going to be talking about are called NAD and FAD. So what these two stand for is kind of irrelevant. We won't be required to know what NAD and FAD stand for. But what we do want to know is that they have high affinity for electrons. So they are going to be reduced. And when they do that, when something is reduced, when it's picking up electrons, oftentimes it's not going to be simply picking up electrons. It'll also pick up a hydrogen atom. That is how things are often reduced in the body. So what happens is you have this electron that's going to be picked up by NAD. I usually write electron as just E- for shorthand. So because this atom has a high energy or a high affinity for electrons, not this atom, but this molecule has a high affinity for electrons, it's going to pull that electron toward it. But in order to stabilize and let it carry that electron, it's also going to have to bring in a hydrogen. So what's going to happen is NAD picks up an electron and it uses that electron to form a new bond with hydrogen. So during the process of reducing or adding an electron to NAD, you're forming this molecule NADH. You're just adding the electron and this hydrogen to NAD to make NADH, which we would say has then been reduced. Same thing happens with FAD, except that FAD is going to tend to pick up 2 electrons and 2 hydrogens at a time. So we're going to say FAD is being reduced, it's adding 2 electrons, but it's also going to use those electrons to form 2 new bonds with hydrogen. So when FAD is reduced, what you're left with is FADH2. So these electron carriers, they're picking up these electrons, and where they're getting them from is going to be when you're breaking the bonds of other molecules. So when you remove an electron, oftentimes to get that electron, you're going to have to break a covalent bond. And what sorts of molecules have lots of covalent bonds that can be broken? Well, things like glucose, the things that you eat, the molecules that you get in your diet, they're going to have lots of bonds that can be broken, and those electrons can be pulled off of those molecules and transferred to these electron carriers. And in that transfer, that's going to give off a little bit of energy. Once these electron carriers transfer those electrons to the next step, you'll see that even more energy is going to be produced. So these electrons, forming these electron carriers, reducing these electron carriers, you'll see in a second, is going to be important for the ability to generate ATP. So right now, I want to explain where the ATP is coming from. I kind of want to jump to the end. So there are a few steps that I'm skipping right now, but I think that since we've already talked about these reduced coenzymes, the NAD and FAD, they've picked up electrons, they've been reduced, and now they're carrying those electrons. I kind of want to jump to the point a little bit and tell you why they're doing that. So where these reduced coenzymes, NADH and FADH2, are going to end up is inside the mitochondria. So the mitochondria itself looks something like this. And it's got two phospholipid bilayers. It's got an outer membrane and an inner membrane. And inside that inner membrane is a space called the mitochondrial matrix. And then in between these two membranes is a space called the intermembrane space. So in between those two phospholipid bilayers, in between those two membranes, is the intermembrane space. Let me write that. And everybody's heard the mitochondria is the powerhouse of the cell. The reason for this is that it's inside the mitochondria that we're producing ATP. So carrying electrons around is clearly, I've placed emphasis on that because that's the most important part of ATP generation. The vast majority of ATP that's produced is produced by transferring electrons. So kind of jumping to the point, inside the mitochondria, on this inner membrane, so this is the inner membrane that I have right here. In this inner membrane, you have these proteins, these transmembrane proteins. Remember, we called those integral proteins that are embedded in that membrane. And we have these electron carriers, NADH and FADH2. They're going to be transported to this inner membrane inside the mitochondrial matrix. And they are going to release those electrons at this inner membrane. So NADH is going to be oxidized. It's now going to lose that electron that it picked up. It's going to lose that electron, and that electron is going to be transferred to one of these proteins in the mitochondrial matrix. And remember, an electron moving gives off some energy. But that energy is not going to be used yet, not just yet, to make ATP. What these proteins that are taking that electron are doing with that energy, as that electron bounces around this protein, it's going to use that energy to pump hydrogen ions. So you have a hydrogen ion here. That electron is moving through this protein. It's releasing some energy. And this protein uses that energy to take this hydrogen and push it to the other side of this membrane. So you have these positive hydrogen ions. And the same thing happens as that electron bounces through these different subunits. So this electron is dropped off here at this first subunit, this first protein. An electron bounces around again. Hydrogen gets pumped to the other side of this membrane. So now hydrogen's here. Same thing happens with this. And all those electrons moving around in this inner membrane, they're releasing energy that's used to pump hydrogen ions. So again, you get this gradient that's being built up. Lots of positive ions fill this intermembrane space. And you're just building up this larger and larger, this steeper and steeper concentration gradient of hydrogen ions. And the purpose of that is that the more of these ions that build up on one side, the more they want to diffuse back into the other side of the membrane. But they're only going to be able to diffuse through one special protein in that membrane, and that's called the ATP synthase. So what this last enzyme is in this membrane, this last protein, is this going to be a channel that allows these hydrogens that have built up here, lots and lots of hydrogens have built up here, they are going to diffuse back through toward their concentration gradient. That's where they want to be. They want to bring equilibrium. So hydrogen is going to move so that there will be equal amounts of hydrogen on both sides again. And as hydrogen moves through, it's going to turn what's actually a molecular turbine. So this last protein here that hydrogen flows through, ATP synthase, it's functionally equivalent to a turbine in a hydroelectric dam. So what happens in a dam? You have lots of water on one side of the dam, not as much water on the other side. And as water flows through that turbine is going to spin and generate electricity. So these hydrogen ions that have built up on this side of the membrane are like the water on one side of a dam. And they're going through this turbine, and as they do, the energy of the movement, energy from the movement of those ions is going to turn this turbine. But instead of making electrical energy, ATP synthase takes ADP, an inorganic phosphate, and every time it turns, instead of making electrical energy again like a dam does, it's going to take that ADP, that inorganic phosphate, and convert them into ATP. It's going to store that energy. So this is what's called a chemiosmotic gradient. So through the process of what's called chemiosmosis, that's just where these ions are moving through this ATP synthase. You've built up this concentration gradient, hydrogen moves through it. That chemiosmotic gradient basically is potential energy. And when that potential energy is turned into kinetic energy, as hydrogen ions move through to the other side of this membrane, they turn a turbine, and that turbine uses that energy to make ATP. So I've skipped the process of how we go about making these reduced coenzymes. We'll talk about that in just a second. But the payoff here is that these electron carriers are carrying electrons to the inner membrane in mitochondria. So this whole process, transporting electrons using the energy from electrons bouncing around to pump hydrogen ions, using those hydrogen ions flowing back into the mitochondrial matrix to turn the turbine and make ATP. This whole set of proteins, this whole process, is called the electron transport chain. Or we abbreviate that, the ETC. So most of the ATP that the cells in your body make is going to come directly from the electron transport chain. Transporting electrons, using the transport of electrons to pump hydrogen ions. And when those hydrogen ions flow back into the membrane, they turn a turbine and generate ATP. So the electron transport chain, again, is where most of the ATP is going to come from. But I haven't described to you yet where the reduced coenzymes come from. So we'll go back into chronological order now and talk about where these reduced coenzymes come from. So you know how ATP is made now. You're basically generating lots of these electron carriers that have been reduced. So your FADH2 and your NADH, and they're taking those electrons to the electron transport chain where ATP is made. But where do those reduced coenzymes, NADH and FADH, how do those get reduced to begin with? And that's through glucose catabolism. So glucose catabolism, of course, starts off with glucose, which has 6 carbons. So 1, 2, 3, 4, 5, 6. Each of these circles will represent 1 carbon of glucose. And the first step in glucose catabolism that's going to ultimately end up making you lots and lots of NADH and FADH2 that can be used to make lots and lots of ATP. The first step is called glycolysis. So if you break down that word glycolysis, it means glucose lysis, which is just breaking down glucose. That's what catabolism is breaking something down. So you have this 6-carbon glucose molecule. And in order to break that molecule down, you first have to add ATP. There's the investment phase. So you're investing 2 molecules of ATP. And inputting glucose, you add those ATPs. And what this reaction is ultimately going to be, the first step in glycolysis, is going to be breaking apart that glucose molecule into 2 3-carbon molecules, each with a phosphate attached. That's why you need the ATP, is to add a phosphate. So this is a molecule called glyceraldehyde 3-phosphate, or G3P is how I abbreviate it. But you see it's got these 1, 2, 3 carbons, it's got the phosphate. So this is your 3-carbon. And since you're splitting this 6-carbon in 2, you actually end up with 2 of these glyceride 3-phosphate molecules when you split apart that glucose. So as we go through glucose catabolism, there's going to be some steps in the reactions that I'm going to kind of skip over. I'm just going to give you the highlights, and these will be the ones that you'll want to focus on when you're studying for something like an exam. During glucose catabolism, generation of ATP, there's a lot of minutia, which is not to say it's unimportant, but I'm not training you all as biochemists. I'm training you to be able to understand how the body produces ATP, and I think you can do that by understanding generally what's going on in glucose catabolism. So again, the first step is glycolysis, and you have to invest 2 ATPs. So the net ATP, at least as far as this step, you've used 2. So you've lost 2 ATP here. So net, you're down 2 ATP. That's what investment means, is you have to spend some money to make some money. So you have to spend some ATP here to make ATP. So what do we do now once we've got these two glyceraldehyde 3-phosphates? Those are going to be converted into a molecule called pyruvate, which is going to be a 3-carbon molecule still. Basically, you've removed those phosphates now. And you're left with this 3-carbon, 2 of these 3-carbon pyruvate molecules. And I say this is the payoff portion of glycolysis because you're actually going to gain 4 ATP during the process of conversion of these glyceraldehyde 3-phosphates to pyruvate. You gain 4. So since you lost 2 to begin with and you gain 4 here, you add those up, and what you're left with is, at this point, your net is now, you're in the positive. You've gained 2 ATP from where you started. So just glycolysis, just breaking apart one molecule of glucose, yeah, you spend a little bit of ATP, but you get back more than you spent. So net at this point is a gain. You've gained 2 ATP total. Now, the fate of pyruvate, what happens to pyruvate, is going to depend on whether or not oxygen is present. So if there is no oxygen, we would call those anaerobic conditions. There's no oxygen present. It's anaerobic, meaning again, no oxygen. If that's the case, if there is no oxygen present, and this might be the case if you're in very strenuous exercise where oxygen is being used up rapidly and there's little oxygen to go around, pyruvate is going to be converted into lactic acid. So strenuous exercise, you're having a lot of muscle contractions. That's why a lot of lactic acid builds up, is because you're lower on oxygen than you normally are. And that pyruvate that you're building up as a result of meeting the energy demands of contracting muscle, they use a lot of ATP. And to get that ATP, they break down glucose. And you end up with this pyruvate. And again, if oxygen is low, that pyruvate gets converted to lactic acid. And it's going to be transported to the liver to be metabolized. Now, if there is oxygen present, that's where you're really going to win as far as being able to generate a massive amount of ATP. So in cases where there's strenuous exercise, you're probably not going to go all the way through to the electron transport chain as much because you're lower on oxygen. But when you're resting, you're going to be able to produce lots and lots of ATP, and that's going to be through the aerobic pathway. So now you've got these 3-carbon molecules of pyruvate. You've got 2 of those because you started off with 1 6-carbon glucose, you split it in half, and you've got 2 of these 3-carbon pyruvates. So we mentioned if there is no O2, they're going to be converted to lactic acid, and again, that's an anaerobic process when you're low on O2. One thing that I did fail to mention is that when you produce pyruvate, in addition to generating 4 molecules of ATP, bringing you up to a +2 net ATP, you also generate 2 molecules of NADH. You reduce NAD to NADH. So you've made 2 of those electron carriers that I mentioned before are so important for making ATP in the electron transport chain. So we've already made 2 of those really important molecules. Now, if oxygen is present, if you're not strenuously exercising or if you're not in a low oxygen environment, pyruvate is going to be transported into the inner mitochondrial matrix. So glycolysis is happening in the cytosol, in the cytoplasm. It's happening outside the mitochondria. But once pyruvate is generated, it's going to be transported into the mitochondria where the Krebs cycle can occur. So if O2 is present, you can proceed to what's called oxidative catabolism. And again, the purpose of this oxidative catabolism, is not to make ATP directly, but it's to make as many of these reduced coenzymes, NADH and FADH2, as possible, so that they can take their electrons to the electron transport chain, and that molecular turbine can turn as a result of pumping hydrogen. So oxidative catabolism, again, that means electrons are being transferred in these catabolic processes. What we're mostly referring to is electrons being transferred to NAD and FAD. So again, this is an aerobic process, whereas anaerobic was generating lactic acid with pyruvate. If you have O2 present, then you're going to go through an aerobic process, which allows you to generate much, much more ATP, as we'll see in a second. So after pyruvate enters the mitochondrial matrix, enters matrix, crosses both of these membranes that you see in the mitochondria. Inside that matrix are enzymes that are going to convert pyruvate into a molecule called acetyl-CoA. Now, this is going to occur again in the mitochondrial matrix, and generating formation of acetyl-CoA. This step is usually referred to as formation of acetyl-CoA. I'll write that out again. So formation of acetyl-CoA occurs again inside that mitochondrial matrix. And what acetyl-CoA is it's actually going to be a 2-carbon molecule. But more important than how many carbons it has is where it's going to go, what's going to happen to acetyl-CoA. And what happens to acetyl-CoA? So if we just visualize these 2 carbons that represent acetyl-CoA, it's going to be joined with a 4-carbon molecule. And when it does that, it's going to go through a series of reactions called the Krebs cycle. Now, your book shows what every one of these reactions of the Krebs cycle is, and I won't require you to know every step in that series of reactions, but what I do want you to know is that in the Krebs cycle, again, it requires oxygen to be present for this to occur, for acetyl-CoA to be formed, and for Krebs cycle reactions to occur. But so first step in the Krebs cycle is that you're going to make a 6-carbon molecule, and then the Krebs cycle is going to start breaking these bonds between those carbons. And as those bonds are broken, you're going to liberate electrons from those molecules and transfer them to NAD and FAD to make FADH2, NADH. You are reducing NAD and FAD. And again, these are electron carriers. So how many of these do you make for every turn of the Krebs cycle? You're going to make 3 NADH and 1 FADH2. But remember, for every molecule of glucose, you're making 2 pyruvates. So there are actually 2 pyruvates that are each going through the Krebs cycle. So if you multiply this by 2, because there's 2 pyruvates, you're actually making, for each molecule of glucose, you're making 6 NADH, 2 FADH2. And you're also making a couple of ATPs. So every molecule of glucose in the Krebs cycle generates 6 NADH, 2 FADH2, 2 ATPs. And where do these end up? They're going to be taking their electrons to the electron transport chain. One thing that I did forget to mention is that during this formation of acetyl-CoA reaction, you're also producing 2 more NADH. And you're also producing carbon dioxide. That's another thing that's produced in great quantities in the Krebs cycle, is that each time you break a bond between carbons in the Krebs cycle, you add some oxygens to it, and that forms carbon dioxide. So the reason you're breathing is because you need oxygen for this aerobic catabolism, but you're breathing out also to get rid of all this CO2 that's building up for every time this set of reactions occurs. So let's think about where we are total as far as our net goes. So in glycolysis, you make your 2 ATP, your 2 NADH. In the formation of acetyl-CoA, you make 2 more NADH. And then in the Krebs cycle, you're making 6 NADH, 2 FADH2, 2 more ATP, and some carbon dioxide, of course. So once we've gone through the Krebs cycle, our net is we've gained 4 ATP, 10 NADH, 2 FADH2. So these are our important products of aerobic oxidative catabolism of glucose. For one molecule of glucose, this is what you get through these reactions. Now, 4 ATP isn't actually all that much, but when these 12 reduced coenzymes take their electrons to the electron transport chain, the electron transport chain production of ATP for every 12 of these is going to produce around 34 molecules of ATP. So just glycolysis, you get 2 ATP, and then the Krebs cycle you get 2 more. But it's really the electron transport chain that is going to be the engine of ATP production, and that's why oxygen is so important. And we'll talk about the last step of where oxygen plays into this in just a second. So why again is oxygen necessary for glucose catabolism to generate that extra 34 ATPs that you get out of the electron transport chain? Well, so when these electron carriers, NADH and FADH2, when they donate those electrons and they start bouncing through the electron transport chain, every time an electron moving again, it pumps hydrogens. And eventually those hydrogens move through, turn the ATP synthase to make lots and lots of ATP. The final electron acceptor, so each of these proteins that's part of the electron transport chain is accepting electrons. So it's taking an electron from NADH or FADH2, and they're being passed between these proteins. They're accepting those electrons. The last molecule that accepts the electron, the final electron acceptor, is O2, is molecular oxygen. So that electron is going to finally be accepted by O2. And again, remember when something gets reduced, it gains an electron. Oftentimes a hydrogen comes with it. So that oxygen gaining that electron is going to make H2O. So oxygen gains some electrons, it gains hydrogens, and it's going to produce lots of water. But you need that oxygen to be there for this process to occur at all. If that doesn't happen, then electrons are going to have nowhere to go. They're going to build up, and the electron transport chain won't run efficiently. So oxygen must be present in order for the electron transport chain to work, so that you can get that extra 34 ATP. So I know we kind of went roundabout through the process, but let's just talk through it one last time. So if you've got a molecule of glucose in the cytoplasm or the cytosol, that glucose is going to be broken down through glycolysis. And again, that does not require oxygen. The next step would be formation of acetyl-CoA. And for this to happen, this again occurs inside the mitochondria. For pyruvate formed through glycolysis to enter the mitochondria and get turned into acetyl-CoA, that's not going to happen if there's no O2 present. Once that acetyl-CoA has been formed, it enters the Krebs cycle. Krebs cycle produces lots and lots of NADH and FADH2, and those are going to be processed through the electron transport chain, which is where you get your energy payoff. So now I want to spend some time talking about how this is all regulated. And things that are regulated homeostatically are often controlled through hormones. Those biological variables, lots of different biological variables that are controlled homeostatically. The one that we're going to focus on right now is blood glucose levels. So how much glucose is in your blood? And that's tightly regulated. Too much is a bad thing. Too little is a bad thing. You might wonder why having too much glucose would be a bad thing. You think sugar is something that's good, we can make energy from it, but too much of it can actually do damage to tissues. So too much blood glucose can be toxic, and too little, of course, means you're not going to have enough of that substance that allows you to make lots and lots of ATP, which we need to do lots of things. So keeping blood glucose levels in a specific range is going to be really important. And your pancreas is the organ that controls this. So your pancreas is located right around your stomach, and the reason for that is, because it's an exocrine organ that makes digestive enzymes. So the pancreas makes certain enzymes that help to break things down in your stomach, break down food. In addition to being an exocrine organ, it's also an endocrine organ, meaning that it produces hormones. So the hormones that your pancreas produces, the two that we're going to focus on, are called glucagon and insulin. And these are going to be antagonists to one another. So, and what that means that they have the opposite effect to one another. What glucagon is going to do is it is a signal that blood sugar is too low. So what it's going to do is signal cells to increase blood sugar levels. Insulin does just the opposite. It's going to, the action of insulin is going to end up lowering blood sugar. So before we get into any more about glucagon and insulin, I want to talk about the pancreas. And if you look at the pancreas, it's kind of this organ that looks something like this. It kind of sits right next to your stomach. But if you zoom in on the pancreas, you would see, if you looked at it under the microscope, you'd see what are called islets. Islets of Langerhans is the technical term for them. But what these are is just little patches of cells. So if you took your microscope and looked really closely at the pancreas, you would see these little islets, these little clusters of different types of cells. So I'm drawing some green cells here and some black cells. And we'll say that these black cells are called alpha cells. And we'll say that the green ones are our beta cells. So those are just Greek letters, A and B basically. So these two types of cells, there's a third type called the delta cell, but we're not going to focus on those. We're going to be focused on these alpha and beta cells. So there's these little islets all over the pancreas that produce insulin and glucagon. So the cell that produces glucagon is going to be your alpha cells. And the cells that produce insulin are your beta cells. And so when you think about how a biological variable is controlled by the body, there's a control center, but there's also a receptor. And those receptors are going to be measuring how much blood glucose there is. So in this case, these cells are going to be both the receptor and the control center. So they're producing glucagon or insulin in response to their measurement of blood glucose levels. So if blood glucose is too low, these cells are going to favor production of glucagon. So the beta cells will be less active. They're going to be making less insulin. Now if your blood sugar is too high, then insulin production is going to be favored. And we'll talk in a second about the mechanisms that are used to either raise or lower blood sugar. But these are the hormones that are produced by the pancreas to bring about that effect of either raising or lowering blood sugar. So let's first talk about glucagon for a second, which again is the hormone that's going to it's going to bring about a change that's going to raise your blood sugar. So what is the mechanism for that happening? Well, what happens is glucagon is going to have target cells that respond to this hormone. And cells like your liver and muscle, skeletal muscle, contain lots of this molecule, glycogen. And what you may remember from when we talked about macromolecules or polymers is that glycogen is a polymer of glucose. So it's lots of these glucose molecules stitched together. It's stored glucose. So one really quick way to raise blood sugar is for the storage site of glucose in the liver and in skeletal muscle, for those cells to release stored glucose. So breaking down glycogen. and releasing that into the bloodstream. So that's one way to raise blood sugar, and this is called glycogenolysis. Glycogenolysis is the process of breaking down glycogen. Another way to raise blood sugar levels is to make brand new glucose, to make glucose from scratch, basically. And cells can do this, and this process is called gluconeogenesis. So this is going to happen primarily in the liver. And you're going to start from raw materials such as glycerol that you get from breaking down triglycerides. You can turn some amino acids into new glucose. But so this is a process that doesn't happen as quickly as just releasing stored glucose, but new glucose formation can also happen to raise blood sugar levels. Another way that you can raise blood sugar levels is to break down triglycerides. So part of the triglyceride, that glyceride comes from the fact that glycerol is a part of the triglyceride molecule. And glycerol can be used through this process of gluconeogenesis. So triglyceride breakdown is another way to raise blood sugar levels, but also to account for the loss of your ability to make ATP. So in addition to glucose catabolism, that's your cell's preferred source of energy. Cells prefer to use glucose to make ATP. But if you don't have a lot of sugar around, you can use fats to make ATP as well. So breaking down triglycerides is going to release glycerol and fatty acids. And these fatty acids can be catabolized, same as glucose, although it's not quite as efficient of a process, and what you end up making are called ketone bodies. So some of you might have heard of ketogenic diets, where your body shifts away from using sugar as its preferred energy source and shifts towards preferring to use fats to get energy. And the reason why it's called a ketogenic diet is because that it's going to produce lots of these ketone bodies. Now, ketone bodies have an acidic nature to them. So this can cause ketoacidosis. It'll lower your blood pH if you have too many of these ketone bodies. So that can actually be somewhat dangerous in certain cases. But ketone bodies themselves can be used as another energy source, just like glucose can. So in the case where you are starving, or if you are on a low-calorie diet, or if you're just eating lots of fats and not so much carbohydrates, you can produce lots of ketone bodies, and that'll keep up with your energy production needs. But so these will be kind of the different processes that are going to be stimulated by glucagon release. So again, the antagonist to glucagon is going to be insulin. That's also, again, this is produced by the beta cells in your pancreas, and the effect of insulin is going to be to lower blood sugar levels. So what happens is, if you have some cells, and let's say you have a blood vessel that's got lots and lots of glucose in it. So how you lower that blood glucose level is by causing cells to take up that glucose from the blood. So when a cell removes glucose from the blood, that's going to lower blood glucose levels. So that's basically the primary action of insulin is to cause cells to take up blood sugar. So what they're going to do is in addition to sugar, they're going to uptake all sorts of nutrients. So your amino acids, your sugars, lipids, those are going to be taken up by cells, and that's going to lower blood sugar. If you have those sugars being taken up by cells, they need to do something with it. So most of the time, if you have high blood sugar and you need to lower it, it's going to be because you have either hyperglycemia, some issue that makes it so you can't control insulin levels, but it's going to happen after you eat a meal, so that your blood sugars are going to increase. And insulin is a way to tell cells it's time to take up that sugar from the bloodstream and do something with it. So what are they going to do with it? They're going to do things like synthesize glycogen. If you have high blood sugar, you need to do something with it, let's store it. So that's going to happen in the livers and also in skeletal muscle. So they're going to take, these cells are going to take that extra glucose and store it in the form of glycogen. So anabolic processes are being favored here. Another form of storage, energy storage, triglyceride formation. Triglyceride synthesis. And this is going to take place in adipose tissues. So high blood sugar means you're also likely to have high blood lipids, and so triglyceride synthesis. Again, these are synthesis reactions. These anabolic reactions are going to be favored when there's insulin present. And another effect of insulin is that you're going to feel full. So you are satiated, you are satisfied after a meal, partially because insulin causes you to feel full. So that's a nice effect of having a meal, is you get this boost in insulin production, and your brain recognizes that you feel full. So the last thing I would like to talk about for this lecture is what's happening when blood sugar levels are off. So if you were to graph, if you had blood sugar levels over time, they're not going to stay the same. They're going to kind of fluctuate depending on how long it's been since you had a meal, and also what the opposing effects of insulin and glucagon are. So let's say we're right here. Let's say it's been a while since your last meal and that sugar's been used up, and your blood sugar gets lower and glucagon starts getting produced. That glucagon causes those cells to start releasing new sugar into the blood. Your blood sugar climbs. So your blood sugar levels are just going to naturally fluctuate, just like any other biological variable. But they're going to usually stay within this normal range. So this would be kind of a normal range of blood glucose levels. But there are certain conditions where blood glucose will fall outside of that normal range, whether it's higher, so that would be hyperglycemia, or lower than normal would be hypoglycemia. So you're probably already thinking about what's a disease that I've heard of that relates to blood sugar levels. The one that everyone would of course think of is diabetes. So in type 1 diabetes, it's actually an autoimmune disorder. This is so type 1 is an inherited disorder, an inherited autoimmune disorder where your immune system attacks those beta cells that produce insulin. So you lose the ability to produce insulin. Insulin levels are low. And since insulin levels are low, cells are not going to be taking up blood sugar as they normally would. So you can imagine someone who has just eaten a meal, who is diabetic. As they start to, the blood sugar rises after a meal, it's not going to come back down because insulin production is what tells cells to take up that blood glucose. So they have to take exogenous insulin. They have to inject themselves with insulin in order to bring down their blood sugar levels so that they are not hyperglycemic, so that they are not in the toxic range of blood sugar levels. So you can get neuropathy. Some neurons are going to be injured by having high blood sugar levels. Blood vessels can be injured by having high blood sugar levels. So too high blood sugar is a major issue. In diabetes type 1, you lose insulin production because those beta cells have been destroyed. So what makes type 2 diabetes different is that so you still are going to have the same effect as if you had less insulin. But what happens here more so is the fact that cells become insensitive to insulin. So you can still get insulin production, but the cells that are responding to insulin under normal circumstances in type 2 diabetes, which is acquired diabetes, it's something you're not born with, but you're acquired, you develop over time insulin insensitivity. And because you're not going to have insulin to counter the effects of glucagon, the effects of glucagon are going to predominate, and that will cause the same sorts of issues that you would have if you simply had not enough insulin. So type 2 diabetes mellitus is associated with there's some it's thought that there's some inherited aspects to the disorder, but a lot of it is associated with obesity and your diet. So this type 2 is more so one that you see that you can kind of predict, whereas type 1 is more inherited and is less of a disease of lifestyle. So that's basically it on this portion of our metabolism exercise. And I will look forward to talking to you on the discussion board. I'll be posing a question in the next day or two, and we can talk about some issues related to the metabolic controls and the process of metabolism that we talked about in this lecture. See you next week.